Good morning, everyone. As Anne mentioned yesterday, a formidable challenge in developing regenerative medicine therapies is to figure out what to measure and how to measure it. We believe that the importance of single-cell methods of obtaining high-dimensional cytometric data will be of increasing importance. And this session will try and jump into that in a little bit more detail. To lead off from Johns Hopkins, we have Dr. Elana Fertig, who has recently been doing some very interesting work with alternative methods of matrix factorization. And perhaps she will expound on that in a little bit more detail. Elana, if you would.
Thank you very much for having me here today. I'm going to start by pulling up some slides to talk about single-cell technologies and matrix factorization, as well as other computational techniques that are used for that. Sorry. So let me play my slides. So I'm going to talk sort of broadly about the challenges and sort of what the needs are for developing computational methods for understanding single-cell genomics, and why that's really an important part of the field in addition to the technology. So to start with, it only makes sense in the context of what are single-cell technologies. So if we think about the previous generation of technologies, and we think about things in context of an analogy like, for example, fruit, as I'm showing here, the previous generation of technologies for bulk RNA-seq really were sort of like a smoothie, where we had each of the different components of the system, of a biological system, really blended together, right? So you had all the cells and all the molecules blended together, and you were trying to figure out what was going on. With single-cell technologies, now you're able to pull out one by one each piece of fruit or each cell and measure what the molecular state is. And now where the technologies are heading is not only characterizing what cells are there, what pieces of fruit are there, but really where are they spatially aligned in a tissue. And so it's a very exciting time from a technology standpoint where we're really able to characterize a system in its native context and start thinking about what's going on. So the obvious question that you can get from these technologies is, what are the cell types that are there? And there's been a lot of effort into that, into using single-cell data to really understand the cell types. But arguably, you could have used flow cytometry approaches or other lower dimensional technologies to do that previously. So that's not new with single-cell RNA technologies. So the question is, what is new? So I'm going to give an example throughout a lot of this talk, building from not just, the fruit example, but an actual real-world example. So there have been several single-cell studies. The one I'm going to pull from is one that I did in collaboration with Loyal Goff and Seth Blackshaw, where we characterized the single-cell development of the retina across different developmental time points and different cell types. And so if we think about this data set, and I'm showing here a picture of all of these single cells. So each dot here is a representation of each individual cell in the retina. And there were over 100,000 cells in this particular data set. On the left, we've colored the cells by what time point they were taken from. So these were taken at different developmental times in the retina. And you can see you have this transition over time that you can see in the data. And on the right, we've colored them by which cell type, again, sort of which pieces of fruit. And so these give you two different ways of seeing the system. But again, you could have gone in a priori and characterized these cell types, characterized these ages. So what's really novel? So what Aviv Regev sort of noticed, and what's very exciting about single-cell data, is that because you have the whole transcriptional profile of this system, you don't only get out cell types, but within each of these cell types, you also have an information about what phenotype it's in, what is the temporal progression, where is it along the developmental trajectory, where is it along the cell cycle, and where is it along the spatial position. And each of these are really encoded fundamentally in the data in terms of the set of genes that it regulates. So the question from the data becomes, how do we go deeper than sort of clustering or annotating these cells to figure out what these phenotypes are? And the nice thing about this is that this is a problem that has been answered for hundreds of years mathematically in terms of the problem of matrix factorization. So on the right, I'm going to go away from this complex world of, thousands of genes and hundreds of thousands of cells in a single cell data set to a world that's represented by this cartoon here that I'm showing you on the right, where you have 4 genes and sort of 2 distinct groups of cells. And what you can clearly see by eye is that if you look at each of those 4 genes, there's sort of 2 fundamental types of processes in the data. One that are going up and down in the first set of cells, and one going up and down in the second set of cells. And the last 2 genes are just using different combinations of that. So this idea of matrix factorization is that you can take this data and then learn what are the axes in that data mathematically. And then by linking that back to genes or which cells it's occurring in, you can then link it back to the phenotypes that are observed that are sort of fundamental to the system. So when we apply this to this example of retinal development, and I'm showing that on the right in those previous sort of taking that picture from before, what you can see is that some of the features that are learned through a matrix factorization approach, for example, the bottom right, reflect individual cell types. So for example, we're seeing the neurogenic cell type. But in addition to learning just cell types, which I could have done by clustering before, I'm also learning fates that reflect changes in cell state. So for example, on the top right, what we observe is this transition where part of the cells are occurring in this pattern that is enriched for cell cycle genes. And this is consistent with the desynchronization of the cell cycle as cells go from being stem cells to developing their final states. And if we look across all the different axes of this data set, we're able to tease out all the different cell types as well as these different transitory states related to age within one algorithm. So the inference of cell state, so this gives us sort of one global way of looking at the system. But there's a lot more there, right? So the axes are giving us a sense of what's going on in the system. But even if you learn one cell type, there's going to be additional molecular heterogeneity within that cell type that could define variation that you need to understand. Some of that variation could be associated with cell state trajectories, like I showed in, for example, the example of the stem cells. And then there's also how do we learn these sort of interaction networks and how cells and molecules work together to drive phenotypes. So as one example, so if we sort of think about cell state transitions, I showed you before how this matrix factorization approach is able to model the state transitions. But this is not the only approach for doing this, right? So if I really know I want to look at the dynamic cell state transitions, I might want to go from a matrix factorization approach to another type of computational method that's specifically geared towards understanding these transitions. And there's 2 dominant types of methods for this that have emerged in the field. One is building on this notion of RNA velocity, which is this idea that if you use the single cell, the transcriptional information, by looking at the ratio of spliced and unspliced gene products, you can figure out the relative maturity of a cell and use that to really rank where it's going over time. And that gives you this velocity field through the concept of RNA velocity on the bottom left. The other idea that's commonly used is this idea of trajectory inference or pseudotime, where the concept is, can we look at cells, if we know the cells, sort of the mature, what are the cell types that are occurring in these clusters? Can we look at how cells, their distance away from each cluster, to figure out how they're changing over time and order the cells? So these are all three different metrics of figuring out what are the cell state transitions. And I think this idea of the dynamics and sort of the fundamental time changes in the system that are observed through single cell data are going to become, are going to really move the field through a systems approach of thinking not only in a data-driven approach, but how do you integrate information about time from dynamic models? The next idea is that even if you're within a single cell subtype, we see some of these temporal changes, but there could also be additional molecular heterogeneity. So not everything is going to be using the same pathways at the same level. So it's not that a pathway just turns on and off. It could be that it's more variable in one state than another. And so there's another set of approaches, for example, our EVA method, that lets you quantify what is the dissimilarity, how different are transcriptional signatures between one cell and another within the same state. And again, if we think of this case of the cell cycle that's sort of showing up in early time points in stem cells, the question is, why is it going away? What we find is that if we look at those same stem cell genes over time, in terms of how dissimilar their profiles are, how much they vary, we find that increases over developmental time. So it's this idea that you're not seeing it because you have more diverse cell cycle states within the mature system, rather than it being that the cell cycle is just turning off. So the next concept is that of regulatory algorithms. And you really need to think about, like, what is driving these transitions, right? Why is the system getting dysregulated? What are the molecular networks underlying that? And there's two sort of dominant approaches that are emerging in the field to start thinking about this. One is this idea of, if we know the trajectories or how the system is changing over the time, can you use the information about which genes are coming up when over time to figure out a network of a sort of causal network of what's occurring when and how that occurs over time? The other major concept that's emerging is if we have prior knowledge about how gene regulation works, for example, through ligands and receptors triggering a cascade to transcription factors, can we then link that molecular knowledge with the data to figure out how the molecular networks are being turned on between cell types? And I think this is really one of the very exciting and emerging areas for single cell technologies, is I think there was a lot of attempts in the early bulk days to figure out these regulatory networks, but they were confounded by these intercellular interactions. And these data give us a new way of really discriminating the inter and intracellular interactions. If we start thinking about that too, not just in terms of inference, in terms of the network, what's happening in the field now is we're going from a point where we just have dissociated cells in single cell to fully spatially resolved single cell data. So this is a data set that I got in collaboration with 10x Genomics with their new Visium platform of a breast tumor. And you can see on the left, if you use pathology, it's annotated into several different regions. And then each of those regions where I've shown you the picture in the slide, you have full transcriptional profiles that are spatially resolved. If you apply the same matrix factorization approach that I showed earlier to figure out the axes of cellular identity to this data set, now we're able to start distinguishing the regions in a de novo fashion to figure out what the dominant signaling processes are that are distinguishing the different tumor regions. And we can also figure out the same thing in the immune system. So in the middle, I'm showing you the patterns that come up that are associated with the tumor that we can then link back to distinct molecular pathways. And on the right, the patterns that are learned with the immune system that you can overlay to start figuring out directly from the spatial interactions how these systems interact. So you can imagine going back to these network inference information these network inference algorithms, and now having information about space as well as time and regulatory networks to really build up what is the potential of the system to be interacting and where are their physical interactions. The other exciting thing that I think is coming up with these is that with single cell technologies, because of the flexibility of the barcoding system, a lot of previous technologies really limited us to transcriptional information to study regulatory mechanisms. The very exciting thing about single cell technologies is now we have these systems where we're able to characterize information across multiple molecular scales, align them, and figure out what are the regulatory networks that, say, arise from DNA alterations that then drive those transcriptional changes. So obviously, I've sort of highlighted a lot of work that was done in my lab, but this is not, this is a very broad and exciting field of thinking about, starting to think about these approaches and really what single cell algorithm development is going to require in terms of matrix factorization and latent space analysis. How do we study cellular heterogeneity? What's the trajectory and velocity analyses, and what are the network analyses? So I'm happy to take any questions. Thank you very much.
That was absolutely fantastic, and I very much appreciate the overview. I think we may wait for questions until all three speakers have presented, and I'll be getting them from the audience as we begin. I'll give you a preview, though. The concept you suggest of collapsing gene space into signaling network space may provide a whole lot of opportunity to simplify questions of cell state characterization in a way that would be useful to developers of regenerative medicine therapies.